So today I am talking about the molecular approach towards the reversal of chemotherapy resistance in high-grade serous ovarian cancer. So what is high-grade ovarian serous cancer? You people already know for that, so I will skip that. So I will skip those these slides. So I am directly here. I am conducting is ongoing project. So I will share some observation what we find. So this is the TCGA database analysis. So here mostly you know that P53 is the highest mutation. P53 is the topmost mutation even in all cancer for our high-grade ovarian cancer. So that is, but if you consider from the TCGA database, there is no such kind of mutational difference in between resistant and sensitive pattern of these patients. So in our institute, what we did that we are conducting a project regarding that how to overcome the resistant pattern of this thing. So we considered that 15 high-grade ovarian serous cancer patient go for resection and nucleic acid extraction, go for exome sequencing and RNA-6. All the patients received 6 cycles of carboplatin adjuvant chemotherapy and for 40 months follow-up data. So 5 will recur within 12 months and 10 recur after 12 months. So we considered that for our analysis. It's still going on for mutational analysis. So far, we found that all the 15 patients are P53 mutant, and 4 are that BRCA mutant. From them are 3 are BRCA mutant, and BRCA2, and 1 is BRCA1. But interestingly, those BRCA mutant are not falling in the early recurrent of the cancer. And here, and in the transcriptomic analysis, we are analyzed and we found that 1768 are upregulated gene and 57 are downregulated gene. So we go for a heat map situation. If you see that yellow one is represent that early resistance and green one, that yellow one represent that mutant and green one represent the early late recurring patient. The thing is that you find that all these, it is very heterogeneous population. If you find that even in gene expression point of view, and then we go for upregulated genes. We go to the gene ontology program and see that most of the genes are from secreted one, and they are located in the also cell membrane. So then what we can do, that how we shortlisted our result. So we follow that whatever database is presented in there, the same kind of treatment they are receiving, and find out that gene expression profile. So we found that these two data will match in our experiment. So we go for that, but they are that microarray based analysis. So we got that expression of this thing. So we combinatorial analysis for this thing. So this is our setup. So we found in the downregulated gene, there is no common findings is there. and from upregulated gene we found in only 7. These 7s are commonly found. In between that, MMP8, tennessine C, BIRC7, that is a live-in that called also, collagen 11, FGF4, MMP10, and collagen 6. Most of them are secretory protein. So next we go for that, whether they are involving for resistant mechanism or not. We go through that literature survey. We found that collagen 11 and this are responsible for that resistant chemo-resistant pattern. So we found, these are the literature review. We found that collagen 11 also represent that chemo-resistant pattern. We also found that collagen 6 also, there are some reports regarding the collagen 6 also. We found that BRC7 is live-in, so it is also responsible for, it is an anti-apoptotic protein. So it is an inhibitor of apoptotic protein. So it is also responsible for resistant mechanism, basically. So then we proceed our analysis through ISC, and we are selecting those genes, collagen 11, live-in, collagen 5, and SMA. And we running through ISC. This is one kind of for collagen 11 expression. What we find that mostly collagen 11 expresses in the tumor cell. In generally from the literature we found that collagen is secreted only through the fibroblast. But here we found it is happened in in tumor cells are mainly express this thing. So this patient is recurred within 10 months. So we go for that live-in expression. It is obviously it is expressed in the tumor cell mostly, and then we found that SMA is mostly expressed in that stromal region, and this is collagen 6. If you find that is expressed in overall as well as tumor as well as stromal cells both. Then we compared with other, that patient 1 recurred within that 28 months. You see that collagen expression, collagen 11 expression is mostly in the stroma, not so much in the tumor. And also live-in is not expressed, and SMA is mostly stromal expression, and also this one is also the stromal expression, both in collagen 6 expressed both. This is another patient recurred in the 6 months. You find that also that collagen 11 is expressed in the tumor cell mostly, but not in the stromal cell. So I skip all this data, and then we compared this. We analyzed 65 patients with a 40 months survival overall recurrent information. So we found that in collagen expression expressed in the tumor region has a poor prognosis, and whatever expressed in that stromal region, they are good prognosis. What we find, and also that live-in expressed in the tumor region is the bad prognosis. So we go through that recurrence-free survival analysis for the 40 months. We found that this is the result, and it is very significant. In the collagen stroma, this thing, and collagen 5 is not related to responsible for in the stromal region, and SMA also, whereas we found that tumoral live-in is also responsible for bad prognosis. Then we surprised that mostly this collagen is expressed, so far that literature also shows that collagen is expressed in fibroblast. How it is, what we are finding, it is totally different. So we go through the again literature analysis. We found this in esophageal cancer, it is expressing the collagen in the tumor region is a bad prognosis. Whereas also this breast cancer malignant tissue also, these two literature shows that collagen 11 expression in that tumor, specifically the tumor region, is a bad prognosis. Whereas in stromal region, it will be some kind of good prognosis. So we go for that collagen and lipin expression through the correlation part. It is highly correlative. Now we are thinking that whether it will be that coincident happened together or is there any interrelation finding is there. So we go for the in silico transcription factor search, and we find that one protein GBPA is a transcription regulator, is a binding together in the both side in the promoter region, and we found that position, and it is that it predicts a good prognostic factor, that GBPA. When the GBPA level is low, then the tumor will be more poor prognosis. When the GBPA level is high, then it will be good prognosis. So we go to our sequencing analysis. So we found that here we also finding the GBPA which is correlated with data that down-regulated gene. Then we look out at what about in the publicly database the knockdown of the GBPA, whether it will go upregulated or not. So in the database in the prostate cancer cell line, we found that if shRNA mediated knockdown, that collagen 11 and lipin are highly expressed. Then we go for the TCGA database and see that is there any correlation in that collagen and lipin expression through the GBPA, and we found that it is a negative regulation. Then we go for another publication database where we found that is found in that HEOC cell line, where the plaquettic cell resistant cell line is OV3 and OVCAR3, where this collagen expression is BRCA, that is the lipin and GBP expression is low, and these two are high. Also in the OVCAR plaquettic cell resistant cell line, it is found in there. But there is no significant change in carboplatin, cisplatin, oxyplatin resistant treatment in those cells. Whereas another interesting thing is that non-HEOC cell line do not express this kind of pattern. So already from the literature we found that GBP expression is regulated by that DNA methylation. So, and DNA methylation an important event for the drug resistance. We found other literature also. So from the TCGA data analysis, we found that GBP expression is downregulated through the methylation. And in the progression-free survival, if it is high expression, then progression-free survival will be better. And we go to our, again, the sequencing thing. We found that any methylation gene, what is the DMNT1, is upregulated in our early record patient. So recently we found that another for RNA-seq data set where they are the same kind of treatment they are giving, and they are finding, they are comparing by sensitive versus resistant. So they are finding these are highly upregulated, the DMNT1, the GBPA is downregulated, and BIRC7 is upregulated. Also collagen 11A1 is upregulated. Next we look at that, is that signature is related to other treatment. Like we go for that publicly available database, we found that when the collagen 11 is highly upregulated, so that survival is poor. That progression-free survival is poor. When it is treated with gemcitabine, there is no difference. Again, the topotecan, there is no difference. And that collagen 6 expression is not significantly not regulated with a bad prognostic factor. Whether it's lipin and GBP, GBP is a good prognostic factor, and it is high expression. So then we check from that almost 85 about patient. Here we see that collagen expression which we are differentiating in the late record and early record. In early record, collagen 11 is highly correlate, and lipin, all these things are highly correlate. Also DMNT1 is highly, but not DMNT3 and DMNT3A and not DMNT3. Then we develop that resistant cell line for particularly in platelet cell in our, because we are choosing OFCA3 cell line which is already chemo-resistant, platinum-resistant cells. So we developed that platelet resistant cell line, and we found that when it will increase in that resistant cell line, that IC50 value of that carboplatin is not so much difference in sensitive versus resistant cell line, whereas the platelet axial resistant cell line, the IC50 value is increasing. Then we look at that mRNA expression in that cell line, where we found that GBPA expression is low, whereas collagen 11 expression is high, and ligand expression high, DMNT1 expression is high. But here we found also DMNT3B expression is high compared to that sensitive. Then we saw that what happened, whether we knock down that collagen 11 and GBPA expression. Then we look at that in the OFCA3 cell line, when we knock down the GBPA cell protein gene, we found that that relative expression of mRNA of collagen and ligand is increasing, whereas we found that when we knock down in the collagen 11, that ligand expression is also reduced. So in resistant cell line, we also found this is the same kind of thing. And also, then we look at that platelet cell sensitivity after knockdown the cells. We increase that platelet cell sensitivity in OFCA3 cell line when we knock down the GBPA1 cell and we measure the LDH release assay. So we found when we knock down the GBPA, then we found that these cells are less response in the platelet action. When we knock down the same experiment we repeated in that resistant cell line, we found that when you knock down that collagen 11, so it will be more sensitive to platelet action. Then we check with the cell viability assay with the HUC cell line with the DMNT inhibitor, where we are using two type of DMNT inhibitor. One is decitabine, another one is GSK 48786. So what is decitabine is already established a drug which is used for AML very few, and then GSK inhibitor is specific for the DMNT1 that we are using. So if we are introducing in that in presence of that OFCA3 cell line, we did not that IC50 value is less reduced in that when we treated with decitabine and GSK, but not so much. When we in the resistant OFCA cell line, when we introducing that the platelet cell sensitivity is reduced so. So this is our data. So in our future plan, we have to say that protein expression to the Western blot using the DMNT inhibitor, IMSA to prove that GBPA binding site, and now we are looking for the how to increase the platinum sensitivity. If possible, we can follow the animal experiment and find out how the collagen is regulated, that inhibitory apoptosis protein. So, in acknowledgement, I have to acknowledge Durga is mostly involving all this experiment to perform. I also acknowledge Akhilesh, Dr. Aruna, Professor Kondaya. In the pathology department, all the IC Divya will perform and also help with Dr. Rekha Madam. And I also thank to Dr. Annapurna and our head of the institute, VSS Sinha, and I'll also thank to Dr. Lakshmi Krishnamurthy. Mostly I also thank that Mixed Steel who gave the funding for us. That's it. Thank you.